Alrighty. So before I get started, I'd like to acknowledge and sincerely thank the traditional owners on the lands which I have done field work, and I'd like to pay my respects to their elders, past, present, and emerging. So thanks everyone for coming. Today I'm going to be talking to you about some preliminary results I have from one of my PhD chapters. And in this chapter, I was looking at testing for gene flow between populations of Australia's largest tarantula. So as many of you might know, tarantulas are absolutely massive. So here's a photo of a tarantula and a medium-sized snake up in far north Queensland. So you guys can kind of get a size for just how big they might be. Now, tarantulas are not only huge, they are also quite widespread across the Australian continent. So here's just 5 example species that I've collected from various field trips. And you can see that they occur in basically all of Australia's major biomes. You can find these guys in the wet monsoon tropics and rainforests. You can find them in savannas, dry sclerophyll forests. And remarkably, you can even find these guys in the heart of the arid interior of the Australian continent. Now, tarantulas are super widespread, but a lot of people might not have actually seen a tarantula before. And that's because they typically live a quite cryptic lifestyle. So most tarantula species typically reside in a burrow deep underground, and some other species make use of opportunistic hides, such as tin sheets, old logs, and things like that. Now, tarantulas have a few life history traits that tend to impact the distribution size of any given species. So one of the first major things is that we have non-moving females. So it's thought that tarantulas, once the female spider leaves the maternal burrow, that she'll go ahead and create a burrow of her own, and that she will stay in there for the rest of her life, given that she's not disturbed by or collected by some researcher. And you can just see how much effort they put into these burrows, which can sometimes be about almost a meter deep. And additionally, we also have low dispersing offspring. So the females aren't moving, and then when the juveniles are leaving the maternal burrow, they're only thought to travel a few meters away from that burrow. And there's never been any evidence or people citing that tarantulas have aerial assisted dispersal. So this really leaves it up to the males, upon when reaching maturity, will go off in search of females to mate with. So they seem to have the sole responsibility of maintaining gene flow between disjunct populations. So given this combination of life history traits, tarantulas are considered to be short-range endemics, having relatively small distribution ranges. But I want to introduce you guys to the star of my research chapter, which is Selenocosmia crassipes. So Selenocosmia crassipes is the biggest tarantula we have here in Australia, and it would be the biggest spider, except there's a few huntsmans that beat it for leg length. But in terms of mass, these guys knock it out of the park. So females typically range from a grey to a brown colour, but this colour does change between molts. And then we do get the adult males as such in the bottom right corner, which are a brilliant white coloration. Now, Selenocosmia crassipes is interesting because it has a much larger distribution range than what we would expect as a tarantula. So you can see that from the records of Atlas of Living Australia on the map in the left, this species occurs from about as south as Rockhampton all the way to the very tip of Cape York. And this species makes use of burrows underground, but it also uses opportunistic hides. For example, there was one underneath the tin sheet in the top photo, which was one of the biggest tarantulas I've ever collected. Now, given that most Mygalomorph spiders and tarantulas typically have small distribution ranges, we wanted to know if Selenocosmia crassipes was a single biological species. So we wanted to see if there was any evidence of gene flow being maintained across its distribution range. So obviously the first thing we had to do was get samples. So previous colleagues had collected tarantulas, but it wasn't quite enough to do a population genetic study. And I was fortunate enough to collect several others on a field trip to Cape York. And our final samples ended with 71 individuals from 20 localities, which was almost the entirety of the species distribution range. After that, we used 16S as a barcode in order to identify the tarantulas to make sure that we had the right species. So what you can see in this haplotype network is there's quite a lot of diversity in the 16S mitochondrial marker. And and also that most haplotypes aren't shared among locations. But this is also to be expected given that the females don't move and that the mitochondrial is maternally inherited. So we are expecting to have quite a lot of geographic structuring in mitochondrial DNA. However, I wanted to also note that there is this quite strong break in the genetic distance, and this corresponds to a break between the northern and the southern halves of the distribution. But we'll return to that a little bit later. So given that mitochondrial DNA is only maternally inherited, it doesn't give us the whole picture. So we wanted to be able to get some nuclear data, and we wanted to get some single nucleotide polymorphisms, or SNPs. And we used the ddRADseq approach as it's great for recovering SNPs in non-model organisms. So we had a pretty standard workflow. We did everything in-house. So we extracted the DNA, normalized it, then we ligated and barcoded the adapters and sent them to Novogene for sequencing. However, there was one slight hiccup where the libraries got lost in the mail for 10 days, but luckily they were found and I didn't have to redo all of this lab work for one of my chapters. So with SNPs data, once we got it back, we had to decide which pipeline to use. And there's quite a lot of methods that you can use when doing the bioinformatics. And as a start, we went with the STAX pipeline, where we demultiplexed the individuals and we filtered out low quality individuals and reads. And then as tarantulas don't have a reference genome, we had to map the reads using de novo parameters. And we adjusted several of them to see the effect it had on retaining SNPs. We did some further filtering, removed individuals with 50% missing data, and then we only kept sites that had that were present in at least 80% of individuals, which are pretty standard cutoffs in population genetics. So when that was all said and done, our final data set had 1,230 SNPs, but we did lose a few individuals and a few localities from the filtering. But we took that data and then we did some exploratory analysis, mainly a PCA and an admixture. So we did our PCA and R using the RGIT package, and we get this quite strong clustering into 3 separate groups. And if I draw ellipses on the clusters, these clusters match a northern, a central, and a southern grouping. And it's also worth noting that in the PCA, PC axis 1 and 2 explain over 40% of the variation in our data set. So from there, we went and did a structure analysis. So these are the admixture plots for K2 and K3, in which we predetermined being 2 or 3 populations in the data set. And these two K values had the highest likelihood when we went from K1 to K17. And each bar represents an individual, and the color is the probability of assignment to one of those populations. So when you have individuals with admixture or have had recent gene flow, you can get this ambiguity in which the admixture is struggling to differentiate which population an individual belongs to. But in this data set, we don't have any admixed individuals. So looking at the PCA again, looking at the admixture analysis, we still get those clear separation when we look at K3 between the northern, central, and the southern groupings. So bringing back in our haplotype network earlier on, so we still do recover that really strong genetic differentiation between the northern and the southern range of its distribution. However, while there wasn't that much mitochondrial divergence, we did see further separation in the SNP data between the green and the blue, or the central and the southern populations. So this is only early results, but it does look like Selene-Crosby crassivus could be possibly 3 distinct species rather than a single biological species. And these populations are separated by well-known biogeographic barriers. So to the south, we have the Burdekin River, and between the green and the blue populations, this is, I don't believe this barrier has a name, but it is separated by, it has been shown to separate different taxa in reptiles and amphibians. So this is only the first steps. So we used the STAX pipeline, but we also wanted to use the iPyrad pipeline, which is thought to be a bit more flexible when you have more than one species in your data set. And we also want to do further testing for age of divergence among these clades or taxa. And we also want to do some demographic modeling to see how our data is, how similar our data is to some of the models. and future work looking forward. So this is only the first Australian tarantula species to have SNP data generated, and I believe it's only the second for Australian megalomorphs. So this would be really cool to see what kind of patterns there are for other tarantulas, well, not only tarantulas, but as well as other megalomorphs, given that most megalomorph species often hide cryptic taxa when we do molecular diagnostics. So a big acknowledgement to the Cook Lab and the Regenos Lab here at UQ, which I'm based at. Also a big thanks to Von and Kat for some software troubleshooting. But most importantly, a big thank you to the sampling team, which helped me collect tarantulas on various field trips. So my dad, Von Garcia, Dr. Renan Castro-Santana, and I'm deeply thankful for the UQ Baja Malacu Rangers up in Cooktown, who let me come onto their land, and also they actively helped in collecting samples for this study. So thanks so much, guys, and I'm happy to answer any questions that come up.
Thank you for that, Ethan. Apologies, I had some technical difficulties and missed the start of your talk. But from what I did see, it looks great. Is there any questions for Ethan? So at the moment, I'm not seeing any hands up in the chat. But from what parts of your talk I did see, there seemed to be this group of tarantulas were distributed very in the northern parts of Australia. Do they occur over in PNG as well?
That's something that I'm really keen to look at. So we only managed to get a single individual from a museum collection. But when we look at the some of the mitochondrial markers, they do appear to be sister or in the same clade as sort of the northern group. So potentially they might have dispersed in from PNG, or they might be even connected during LGM.
Fantastic. We have another question in the chat from Peter asking, are you going to look at any morphological differences between locations? Are they potentially 3 different species?
So I think I'm pretty confident about the northern and the southern split, because when you have them side by side, there is a few differences you can notice. Like the ones in the north seem to be a lot bulkier, lack of a better term, when compared to the southern ones. But the sort of the south and the central groups are really hard to tell apart. I haven't sort of noticed any characters, but I would like to follow it up with some morphometrics and things like that.
Mm-hmm. So I think we have time for one final question. Steve has his hand up. Steve?
Yeah, great talk, Ethan. Thanks. I was just interested in how you might distinguish between phylogeographic structure and distinct species when using the SNP data. It's kind of an issue I guess a lot of us have to grapple with.
Yeah, so that one I think is going to be a bit tricky, because if we do detect if we look at the data set, we do find evidence of like sharing of alleles, that might just simply be ILS rather than true gene flow. So I'm hoping that maybe if I'm using the DADI analysis to see sort of a model of where the amount of that might be similar to a high amount of gene flow with like simulated models, so to see if it matches like a deep divergence with limited to no gene flow, or there might be secondary contact. But yeah, I still haven't quite wrapped my head around how, particularly that southern group, how I can separate them out.
Yeah. Okay, thanks.
Cheers, Steve.
Beautiful. So we're right on time then. Thank you for that presentation, Ethan.